The one is called Protostomic development. Protostomic development. This is type of the development which is present in the invertebrates, and this is the development where the mouth is formed first from the blastopore. Blastopore. So what is blastopore? It is the opening of the blastula or the embryo, right? So the second type of the development is called Deutrostomic development. Deuterostomic development, and this is the advanced level of the embryonic development that is present in the chordates and only one phylum of the invertebrates, that is echinoderms, and here the anus is formed from the blastopore. Okay, so what is the difference between the protostomic development and deuterostomic development? In the protostomic development, the blastopore, which is the first opening in the embryo, that becomes the mouth of the animal, and in deuterostomes, that blastopore becomes the anus of the animal, and then the secondary opening appears in the blastula, that becomes anus in protostome and mouth in deuterostome. Okay, so this was the basic characteristic difference between the two, and then there are four or five more differences along the developmental patterns which we are going to discuss now. So if we talk about number one, protostomic development, you know that after fertilization there is a single cellular embryo or the zygote, right, that starts dividing, and then from this one cell there becomes two cells, and from two to becomes how many cells? Four cells, right? So this is a 4-cell stage. Then these 4 cells becomes 8 cells. If I number the cells 1, 2, 3, 4, 5, 6, 7, 8. Okay, so this is called okay, so here you see the cell division is being happening. So in this cell division, you see the cells are dividing and shifting their position, and they are basically rotating along the axis, right? So you see the cells are being shifted in one direction. Each layer becomes shifted into another position. So they, number 1 and number 5 are not exactly on top of each other. Number 2 and 6 are not on top of each other. So this type of the cleavage, the cell division is called cleavage. So this here the cleavage is spiral, right? So the cell division or the cell cleavage in protostomic development, I just type proto here. Protostomic development is cleavage is spiral, and the fate and the type of the cells that are being formed type is equal to determinate. So the type is determinate. So in protostomic development, the cell cleavage is spiral, means that each layer of the cell is shifted or rotated a little bit to give a spiral shape, and the type of the cell is determinate, that the cells, each cell which is formed has its fixed fate. For example, the cell number 2 will make the alimentary canal, so it will always make an alimentary canal. Cell number 4 will make the sensory cells or neurons, so it will always make a neuron. It cannot be differentiated into anything else. So at the time of the embryonic development, each and every cell has its known future and known fate. So this type of the development and cleavage is called determinate cleavage, right? So this is the first characteristic, two characteristics. Then these cells becomes 32 and then become arranged into type of the layer, right? So this is the ectoderm layer is formed first for the first time. So these are all the cells are arranged in the ring-like structure with an empty cavity within, that is called blastocele, right? And this thing is called blastula, and then the blastula that becomes an invagination in the cell outer layer, The cells from the bottom are pushed inward to make a cavity, and they differentiate into a second layer. So this second layer is called the endoderm. And what layer was this? This was ectoderm, right? So at this opening or cavity is called archentron, right? This cavity is called archentron, and this opening is called. So at this stage, you see the animal is diploblastic. It has two layers, the ectoderm and the endoderm. It has a cavity in between that will further along the development becomes the alimentary cavity, and this blastopore will become the mouth of the animal. ठीक है. अब this next step क्या होगा इसमें फिर के we need to have a third layer to establish, that is called mesoderm. Exactly. Okay, let me clean. Okay, let me draw it a bigger image so that you can understand everything clearly. Just hang on for a second. So until now we have seen the gastrula in this form, which has an ectoderm and an endoderm, an archentron and a blastopore, right? And then the third one is that what happens that few cells from this part of the endoderm split up and divides quickly in these empty spaces in this cavity, right, on the both sides, and they are differentiated as the mesodermal cells. Okay, they start differentiating quickly and they fill in the whole space in between the animal's cavity, right? So these are all mesodermal cells are filled in, and then what happens that after this all space is filled in, there is a planned death of the cells in between, in the middle of the middle of these mesodermal masses, and these all cells gets arranged in a way that they make the endodermal layer. So they basically line only the cells which are lining this cavity, they stay, and the rest of the cells in between, they are all dead and they are disintegrated, leaving an empty space. Okay, and then this empty space is called body cavity or coelom. So this is about the coelom formation. Here you can see that the animal is a triploblastic, has an ectoderm layer, endoderm layer, and a mesoderm layer, and an empty cavity between the mesoderm, and that forms the body cavity or the coelom. This type of the development, where the cells were differentiated from the endoderm from this part, and then they filled in the space, and then later the cavity was formed, is called schizocilous development. Schizocilous type of the development, right? And this part, and later on once the development continues, then what happens that a secondary opening appears from here, and that becomes the anus, and this blastopore becomes the mouth, and the development completes, right? So this was all about the protostomic development. So to recap, you have to remember that cell, first of all we will discuss the cell division. The cell division is spiral, and the fate of the cell is determinate. So we can say protostomic development, the animal has a spiral determinate cleavage, then they are triploblastic and they have a body cavity and has a schizocilous cavity development or schizocilous developmental pattern, where the cells from the endoderm split up first, starts, divides, and then makes the cavity later. And finally, the blastopore becomes the mouth, and the secondary opening along the body becomes the anus. This is called the protostomic development. I hope you might get the idea of this type of the development. Now I will not draw the developmental pattern for this for Deutrostomic animals because it is quite similar, and I will show the figure diagrams, and you can easily understand what I'm talking about after watching this drawings. Right, so here you go. That's the main difference and main image that you will see and you can understand here. So the blastula has the cells layered to form layered into to form the ectoderm. So in protostomic development, then these cells from the bottom, they are pushed inward to make the endoderm, right? And a cavity, archentron, and an opening blastopore. Some of the cells from the endoderm split separate from there into the cavity and starts filling in to this cavity and then becomes hollow from within to make a body cavity, a coelom, and then a secondary opening appears into the animal's body and that becomes the anus and blastopore becomes the mouth. In the deuterostomic development, something is different that the blastula are the similar in the both types of the development, but in the blastopore, after the blastopore formation and archentron formation, the cells from the endoderm does not split from it. Rather, they just differentiate as the endoderm, as the mesodermal cells, and start growing and making cavity along while they are still the part of the endoderm. Once they make the cavity and big masses, then they get separated, split up from the endoderm and increase in size by dividing and makes the cavity. Blastopore becomes the anus first and mouth becomes the secondary opening becomes the mouth. Right, so this is a major difference. And if you see difference along with the cleavage, so we just have studied that protostomes have spiral cleavage and determinate fate of the cells. However, deuterostomes have radial cleavage and indeterminate fate. See, the cells are dividing and being placed exactly on top of each other, and they are indeterminate means that the cells, each and every cell has a similar composition, a similar structure, and they are not decided to make anything yet. So they can be differentiated into anything they have to following the command of the DNA. Okay, then the coelom formation, we see the protostomic development is called schizocilous development with the solid masses of the mesoderm splits to form the coelom, whereas in the deuterostomic development, the coelom formation is called enterocilous formation. So enterocilous means that cells differentiate within, entero means within, so within the part of the within being the part of the endoderm, and then they split separate and makes the body cavity. And finally, the blastopore becomes mouth in protostomes and anus in deuterostomes. So that is about the embryonic development and difference between protostomic development and deuterostomic development.